Hello, welcome back to another day, another adventure. Let me just check my mic real quick. Yep, it works. Okay, so right now we are going to talk about DNA topology again, but it'll be focused on the linking number. So, okay, let's start. So, the linking number describes how many times two DNA strands wrap around each other. And what does that mean? It's like a property of closed circular DNA. Just imagine the telephone cord again, just going around. So the linking number can be broken into two components. So let LK be linking number. The two components are writhe and twist. So for twist, wait, let's just so linking number is twist plus writhe, okay? Or writhe. I don't know. I will just say writhe or writhe. One of those could be correct. They could both be wrong, but the spelling is Writhe. Okay, so twist measures how tightly the strands wind around the helical axis. So that's how tightly they are wound. Meanwhile, writhe is the coiling of the helix itself. Okay? So link is how many times, linking number is how many times two DNA strands wrap around each other. Oh, let me check something really quickly. Does this work? If I click up, 2, 3, 4, 5, 6, 7, 8, 9. Okay, it works. So after about like a few seconds. Works after 5 to 7 seconds. 1 second. Okay, there you go. All right, going back to the DNA. Yeah, so it's linked. Linked equals twist plus writhe. So, and twist is the number of times the two DNA strands. It is like how tightly the strands wrap around. And it's about one twist. Like, again, wait, one more second. It's approximately, in the B form DNA, it's described by Watson and Crick, right? It's about one twist per 10 base pairs. Normally, one twist per 10 BP. But in physiological conditions, of course, it's more complicated. It's 10.6 base pairs per turn. So in physiological conditions, it is 10.6 BP. And by physiological conditions, that would be 0.15 molar NaCl at 37 degrees Celsius. So meanwhile, the strain can lead to writhe or writhe or supercoiling to relieve the energy imbalance. So that's why the twist and writhe are like interconvertible. And that would lead to the linking number. And writhe is like how many times the whole double helix cross each other. Interwound writhe is where the DNA helix loops and twists around itself, commonly seen in circular DNA. Toroidal would be where the DNA coils around the proteins. So interwound itself. Toroidal is with other stuff. Again, it is interwound and toroidal. Okay. Again, link equals twist plus writhe. Linking number is the number of times the one strand wraps around each other. Twist is the number of times the whole strands cross each other. Writhe is supercoiling of the whole DNA helix. That's for the telephone. The link of the telephone. And how does supercoiling contribute to the DNA packaging? That's the important thing. The DNA supercoiling is essential for compacting the long DNA molecule into a small circular space. And so again, like, you have to keep on coiling it and coiling it until it is now super wound. And you have in the left picture, which is not shown, there's the unwinding of the DNA by 2 turns, and that would lead to you unwound it by 2 turns, so the twist lessens by 2, so 14 into 12, I mean. But you have to maintain the linking number. So how do you do that? You compensate with the red. It's also -2. So you have a +2 and you have a -2. So it's the same. So let's see. We've said that. Next page. Okay, topoisomerases 1 and 2. What do they do? Okay, so topoisomerases, those would change the linking number of the DNA. How? 1 would break 1 strand. Meanwhile, 2 would break 2 strands. That's the main differences. So how does it do that? It cuts the DNA's sugar phosphate backbone. How does a topoisomerase work? It cuts the DNA's sugar phosphate backbone. We said that previously in a different episode, that the DNA, deoxyribonucleic acid, is composed of sugar, a nucleotide, and that's the nitrogenous base, and sugar, phosphate, and nitrogenous base. And topoisomerases are very essential for DNA function and are targets of many anti-cancer and antibiotic drugs. And as mentioned, they change it by the linking number and break up the sugar phosphate backbone. And after you cut that, the DNA becomes very topologically different. So that's now a new structural state with a new linking number. And yes, that's important. If we go into the specifics of type 1 and type 2 topoisomerases, for type 1, you break the one of the 2 strands by you allow the unbroken strands to rotate and relieve the strain. It does not require ATP, and uses the energy from the broken phosphate bonds to reseal. Meanwhile, before we finish, upon cleavage, the 5' phosphate end of the next strand is linked to the tyrosine residue and the topoisomerase enzyme. So how does type 1 work? It cuts one strand of the DNA helix. Cut. And then once we see, it will twist a bit to relieve the pain, the strain. And then note, upon cleavage, once it gets cut, the 5' phosphate end would be linked to a tyrosine residue in the top isomerase. And that will really make sure that the DNA strand remains tethered while it is trying to like go back to its quote unquote original position, but it won't be able to. And the free 3' end of the DNA is passed through the duplex, and that's a movement it would adjust the DNA topology. There will be a picture like that. Meanwhile, type 2 would cut both strands, change the linking number by 2, and can convert a positive coil into a negative supercoil. Because let's say you have a positive supercoil plus 2, or plus 1, let's say plus 1. And then if you cut both and minus 2, so 1 minus 2 is negative 1. It's a negative supercoil now. Okay. So now we have a mini figure of it. So remember, once you cut one for top isomerase 1, you cut one strand, the 5' phosphate end would be would remain attached to let me just write that 5' phosphate and would remain attached to the tyrosine residue of our top isomerase. And then both strands would try to adjust to the break, a single strand break. And then, yeah, the linking numbers changed by 1. You know, top isomerase 2, both, you cut it, and then would try to adjust, interlinked, and you now have your thing. So replication requires top isomerases. We're gonna talk about DNA replication in a little bit, but yeah, just note that replication requires top isomerases because to relieve the supercoils, and it relieves the supercoils because that would cause torsional strain. Remember, you have the helicase, which opens the DNA helix. And take note, if you try to open up a telephone wire, then where does the remaining energy go? To the other side. So you have the top isomerases to relieve that. Type 1 again cuts a single strand. Type 2 is more powerful. It cuts both strands. And without top isomerases, DNA replication would stall due to the accumulated tension. And I just want to mention some details about a certain journal. So this journal talks about DNA gyrase. It is an enzyme that introduces superhelical turns into DNA. And note, DNA gyrase is a topoisomerase. So the background is DNA, particularly circular DNA, needs to be in a certain supercoiled state for processes like DNA replication and transcription to occur. And DNA gyrase is a type of topoisomerase that introduces negative supercoils into DNA. And supercoiling refers to the twisting again. Refers. One of those. So, well, the problem is, even though we know that DNA gyrase, this was way back then actually, like 1976. So a long time ago. The researchers needed to clarify whether DNA gyrase's action on DNA was ATP dependent and how exactly does it introduce the negative supercoils into the relaxed circular DNA. And there's also a question whether it was a single molecule enzyme or it needed other proteins. So what did they do? They purified DNA gyrase. So how did they do that? They purified DNA gyrase from E. coli and confirmed its activity in converting relaxed closed circular DNA to negatively supercoiled DNA. They used ATP and magnesium ions as a cofactor, and the enzyme activity was assayed by measuring changes in the superhelical density of the DNA. So they purified DNA gyrase from E. coli, and they double checked its activity in converting relaxed closed circular DNA to relieve the negatively supercoiled DNA. And this enzyme activity was then assayed by measuring changes in superhelical activity or density. And then you characterize by the supercoiling, you examine the reaction mechanisms. And yeah, so what they found was they found that the enzyme activity. So the authors confirmed that DNA gyrase did indeed introduce negative supercoils, and it required ATP and magnesium ions, and the enzyme required ATP and also spermidine, aka a specific polyamine, to stimulate the reaction. And if you inhibit ATP or spermidine, the activity is lessened. And it's also a single enzyme without the need for other DNA binding proteins, but it needs magnesium ions and other factors. They have a couple of figures, like 1, 2, 3 or something. So for the first figure, we have, this is a gel electrophoresis, okay? The gel electrophoresis, here they did it on DNA to check the DNA gyrase and its ability to supercoil the DNA. You have a couple of strands, okay? In bands. In lane A, that is supercoiled called E1 DNA without the enzyme. And that's just the starting DNA. It's very supercoiled, it's very tiny, because in gel electrophoresis, the way it works is you have the DNA, you load them into a gel, and then there's the anodes and cathodes. Given here, they did not do SDS page, so I'm assuming that the DNA strands are also still have their charges. So it would travel, take note that the DNA stuff that was loaded onto the gel, they have phosphate, so that is, the phosphate ones are still negative in nature. So they would travel from the negative side towards the more positive side. So cathode to anode, if I'm not mistaken. And also there's a note that the tinier strands or sets or bands would travel faster and farther, meanwhile the bigger ones would remain. So here in lane A, you can see if the bands are traveling from top to bottom, a lot of the supercoiled ones are below. Okay, so that's lane A. Meanwhile, lane B to lane J would be, oh, hello. Yes, it is DNA replication and etc. in a little bit, yeah. So lane A was the supercoiled, so it's at the bottom. That's just the normal one without the enzyme. B to J has varying amounts of DNA gyrase. So I think B does not have it. Yeah, does not have DNA gyrase. So it's still very, very relaxed and very big here. Meanwhile, you slowly increase DNA gyrase, and with J being the most amount of DNA gyrase, it's very super coiled now. Okay? So, and that would imply, well, why is it very super? It's like higher mobility. It was able to like go really, really, really go far. And then you have KLMN, just very specific ones. So, for example, K, the lane K, that one did not have ATP, and as you can see, it is still very relaxed, and it was not super coiled. In lane L, the spiridine enzyme also it is an enzyme known to help stabilize or stabilize the DNA during supercoiling, and if you remove it, you don't have supercoiling. And then M, you removed magnesium, and also in the bands you can see that there is no supercoiling, so it remains in the upper end of the DNA gyrase. I mean of the gel electrophoresis. And so comparing that with N, the lane N, that has DNA gyrase in the absence of ATP, and without ATP, DNA gyrase cannot perform it. So yes, you can really see how DNA gyrase needs, it is a single enzyme, but it needs ATP, spiridine, and magnesium. And then you have their second figure, which shows the characterization of the product. It characterizes the product of the DNA gyrase reaction using GE as again, and it is essentially how DNA gyrase affects different types of DNA. So you have a lot of lanes, A to P. Okay, so lane A is just the normal one. You have the E1, E. coli DNA. It's a plasmid that's commonly used in molecular biology experiments, and this is without any treatment or supercoiling, and it's just the control. Lane B, you have a relaxed form, and it's been treated to remove its supercoiling, so that's why it's still up there. Lane C, it's relaxed with some DNA gyrase, so you can see the appearance of supercoiling below it. And then D and F, D, E, F, those are the same 3 samples with EcoRI. So these show that the DNA after digestion with restriction enzymes, EcoRI, and they pop up in a different like ladder or number area. So right now, if it's super not super if it's super not super coiled, very uncoiled, remains up there. If you have the EcoRI, the restriction enzyme, it's a little bit in the middle. And if it is very, very super coiled, it's below. Okay, super down. So it means it was able to travel really, really far. And then you have P and T1. It's another type of DNA, and just showing how a different plasmid behaves in the reaction. So in G, it was very super coiled because it's below, it's at the bottom of the gel. Then you have relaxed version of it. It's not, it did not travel far. And then now you have the relaxed version of it with DNA gyrase, lane I, and you can see how it's now almost at the same area as its normal one. Next, J is the control unaltered state of E1 DNA. That is J, similar with A. And then you have K, relaxed version of it. And L, relaxed with gyrase. So just showing how if you add gyrase, it ends up being super coiled. M and OP. This one's a funny one. So it's the same panel as L, but aka the relaxed call E1 incubated with DNA gyrase. but just adding relaxed and PNT1 DNA or 5 are just different proteins, extending the incubation time or varying the amount of protein added. They wanted to do different things. Okay, last figure is a 3. What is it? You have the density gradient centrifugation. I cannot flip this page. Wait, I do not want to tear it up. Okay, there. So you just have the equilibrium density gradient or centrifugation. Too many letters. In CSCL, ethidium bromide. Ooh, ethidium bromide. This clearly shows that this is an ancient paper because you don't use. I think if I'm remembering this correctly, you don't use ethidium bromide anymore. A little bit in the recent days because it has been shown that to be carcinogenic. Okay, but let's just travel back in time. They have 4 panels. A, no enzyme. Just before the treatment, lacking DNA gyrase, it's very relaxed. B, you have relaxed E1 DNA with 0.24 micrograms of DNA gyrase. So it's causing some supercoiling. C, it is relaxed with more DNA gyrase, so even more supercoiling. And lastly, panel D shows well, they added 3 micrograms of DNA gyrase, so it's higher amount of concentration. It's higher concentration of it, and even more supercoiled. It's just showing how, okay, if you have DNA gyrase, it's very supercoiled. Okay? Yes. So, guess what? We can now move on to DNA replication. Alright, whoop whoop. Because I've been talking about it earlier. Okay. So, just to give a short outline. DNA, well, what's the structure? What is the relationship between DNA structure and replication? But DNA replication relies on the double helix structure of DNA because those 2 strands, they serve as templates, and replication occurs in a semi-conservative manner, meaning each strand contains one old and one new, and this structure would allow for the separation of 2 strands. And then guess what? That's why each strand is used as a template. So what is needed for DNA replication? Okay. Well, that would involve a set of enzymes and proteins, including helicase, primase, DNA polymerase, ligase, topoisomerase, and single-stranded binding proteins, and a couple more. So wait, let me just note that down. And then, okay. So just a mini sneak peek of what each of those do. Helicase would unwind the DNA protein. Primase synthesizes the primer, so that's why it's primase. And DNA polymerase adds the nucleotides. Ligase would join. It's like a tape. It joins the Okazaki fragments. Topoisomerase, wink, wink. Topoisomerase, we talked about that earlier. It relieves the tension caused by the unwinding, and single-stranded binding proteins are like clips to stabilize the single-stranded regions during replication. All right, and there's also the problem with the leading and lagging strand. Leading strand continuously synthesizes the DNA from a 5' to 3' direction. Meanwhile, lagging strand, it synthesizes the fragments in the 3' to 5' direction, but it still makes the final thing as 5' to 3', but it just has to do it piecewise, so that's why it requires multiple primers. There's also the fact that the accuracy of DNA replication accuracy is crucial to maintain genetic stability, and DNA polymerase ensures fidelity by proofreading the newly synthesized DNA, and it would excise the incorrectly paired bases and it would replace them with the correct ones. Voila. And the proofreading activity of polymerase and mismatched repair system would then further enhance the replication fidelity. And note that replication begins at origins of replication, and those are specific DNA sequences where initiator proteins are found, and you unwind it and form replication bubbles. Note, I mentioned this before. It contains the AT-rich regions, which are easier to separate because A2T. It only has 2 hydrogen bonds. And in eukaryotes, the origin replication complex has a key role. And you have telomeres. How to replicate the ends of linear chromosomes? Well, you use telomeres. Well, you have telomeres, okay? And that's the ends of the chromosomes. You have a unique challenge with that because the lagging strand cannot fully replicate the ends, aka the telomeres. And that would lead to shortening of it, and you can see that in aging. So that's after each replication cycle, and that's why you have the telomerase, which is an enzyme that contains an RNA template, and it extends the telomeres by adding the repetitive nucleotide sequences. So it tries to compensate for the loss of DNA at the ends of the chromosomes during replication. Yes. Okay. So again, as mentioned, just going deeper into the concepts. Or you know what? For now, I think we can end it there. I shall return. But yes, that is all for at this moment. Bye.